So good afternoon, everyone, and welcome to the Replace educational webinars. So for those of you who don't know the Replace project, so we are a platform and we collect methods that can help to the replacement of animal testing, also called new approach methodologies or NAMs. And as part of our project, we organize these webinars to disseminate information on NAMs, but also to feel free to let your email address and name in the chat, and we will provide the certificates in the coming weeks. So that's it for the housekeeping rules. So we can now move on with the first presentation of today, which is a presentation of Nanci. She will present her work on unraveling hepatitis E virus replication in the gut using human intestinal enteroids and a gut-on-a-chip model. Dr. Nanci Ferreira holds a bachelor degrees in bioanalytical sciences and a PhD in biomedical sciences. She is currently a postdoctoral researcher at the Pathogenesis of RNA Viruses and Therapeutic Options lab at the KU Leuven, where her work focuses on using stem cells-derived organoid models and organ-on-chip technology to study enteric virus biology and advance antiviral development. So Nanci, the floor is yours.
So I will start by sharing the presentation. Can you confirm that you see the presentation?
Yes, we can see it.
Thank you. Also, thank you so much for the introduction and the invitation for this webinar. Our lab, the Portal Lab, we studied several RNA viruses, and we use two main new approach methodologies to do it. There are human intestinal organoids and also other human organoids and zebrafish larvae. My work in particular, I focus on gastroenteric virus using these organoid models. And today I will show you the work I have been doing with hepatitis E virus during the last part of my PhD and now my project during my postdoc. And I will start by introduce why it is important to study hepatitis E virus. So this virus is the main cause of viral hepatitis worldwide. And it has 4 genotypes that can infect humans, but their epidemiology and distribution is quite different. So genotype 1 and 2, they are restricted to humans. They only infect humans. And usually infections with these genotypes, they are self-limiting, but can go to up to 25% mortality in pregnant women. On the other hand, we have genotype 3 and 4 that are zoonotic. So they circulate in animal reservoirs, such as pigs, wild boars, cows, sheep. So the main transmission is through the consumption of raw or undercooked pork products, but also transmission by blood transfusion and organ transplantation have been reported. Infection with these genotypes is usually sporadic in high-income countries, but can also cause chronic infections, especially in immunocompromised patients. And the main issue is that we don't have a specific antiviral treatment to treat the infection with this virus. The current off-label options that are used to treat chronically infected patients, they are often not effective in clearing the virus, or they are not suitable for the clinical state of the patient. There is also a vaccine, but it's not available worldwide and also doesn't protect against all genotypes. So this virus is an RNA virus, and it exists in 2 different forms within the human host. It exists in a naked virion, but also a quasi-enveloped virion that contains this lipidic layer. These naked virions, they are very resistant to this infection, and they are very resistant in the environment where they are usually found. And when a person ingests food or water contaminated with these naked virions, they will reach the gut, and then they will cross the intestinal epithelium, and they will travel to the liver where they will cause disease. The specific mechanisms that of this dissemination are not fully known, but we know that the virus is going to replicate in the hepatocytes, and the hepatocytes are going to secrete quasi-enveloped particles into the bloodstream that can either reinfect another part of the liver or can infect other organs, causing typical extrahepatic manifestations that we see with these infections. These quasi-enveloped particles are also secreted in the bile ducts together with bile acids, and it is thought that this lipidic layer of the quasi-enveloped particles is degraded by detergents and proteases that are in the bile acids, explaining why we find naked particles being shed in the feces of the patients. So as you see, there's a lot of the HIV life cycle that we don't know exactly yet how it happens, because this virus is extremely hard to cultivate in vitro, and also in vivo it's difficult, but in vitro it replicates to very low levels, it takes a long time, and on top of that, HIV biology, the knowledge that we have now, is skewed by the use of mainly liver-derived cell lines, such as immortalized cell lines, but also primary human hepatocytes, and now liver organoids. So despite the virus being transmitted through the fecal-oral route, not much attention has been given to the role of the gut in HIV disease. And recently, a group found that HIV is able to replicate in primary intestinal epithelial cells. So this opened the door to a new hypothesis that the gut could serve as a viral reservoir that keeps feeding virions to the liver. And this could play an important role, for example, in chronic infections. So this also then calls for the need to have physiological relevant models where we can study the role of the gut in HIV disease. And that's why we then decided to develop a human intestinal organoid model for HIV infection. And these intestinal organoids, they have paved the way for the development of 3D human organoid models. And nowadays we can almost replicate the entire intestinal, gastrointestinal tract in vitro. But what are these intestinal organoids? So these are self-organized 3D cultures that recapitulate the structure, the cell diversity, and the cell behavior of the human small intestine. And the human intestinal epithelium is organized in crypts and in finger-like protrusions called villi. At the bottom of the crypt, we have intestinal stem cells intercalated with Paneth cells. These stem cells, they proliferate and they keep dividing symmetrically, and they move along signaling gradients that when they reach the top of the crypt compartment, they are triggered to differentiate. And they will differentiate in different mature cell types that include enterocytes, hormone-secreting enteroendocrine cells, mucus-secreting goblet cells, and so on. And these cells, they keep moving along this gradient axis, and when they reach the top of the villi compartment, they are extruded in the lumen. And we see the same structure, crypt and villi structure in the intestinal organoids, with the main difference that in the organoids, we have the lumen enclosed in the 3D structure. So here, for example, you can see the accumulation of the debris and these dead cells inside this 3D organoid. There are also different sources of intestinal organoids. They can be derived from iPSC cells, and in this case, they contain the layer of epithelial cells, but also an extra layer of mesenchymal cells, or the organoids that we now use here in our lab. It's organoids generated from adult stem cells. So tissue that can be collected during a biopsy or a surgery that contains these crypts with stem cells. We can then culture them and create these organoids that are only composed of epithelial cells, and they are usually called human intestinal enteroids to make a distinguish between the tissue-derived and the iPSC derived. So when using intestinal organoids to study microorganisms that infect through the apical side, we need to be able to reach this side with microorganisms. There are different methodologies that can be used for that. For example, microinjection is one of those hypotheses, but it's very laborious and time-consuming. So most frequently, researchers use methods that require the destruction of the 3D structure. Either the fragmentation in smaller fragments that when they close up again, they will contain the microorganism inside, or disruption into a single cell suspension that can be seeded in a monolayer where we have a polarized epithelium and the apical side exposed. So to study hepatitis E virus, we have established different infection models, as I said, using tissue-derived human intestinal enteroids, where we established a model of infection of 3D enteroids, also 2D monolayers, and we also tried the transfection of these enteroids with the genome of the virus. So we began by infecting differentiated 3D enteroids, a method that was also very successful in our lab for other gastroenteric viruses. And to do this, we fragmented the enteroids, we mixed them with the virus in suspension, incubate, then wash and bound virus, and the infected enteroids are plated again in an extracellular matrix, such as matrigel, for example. And we infected 3D enteroids with hepatitis E virus genotype 3, both the wild type, but also one containing a fitness-enhancing mutation. And what we observed was a very limited replication that is very slow over time. This is also a feature that we see in immortalized models. And probably this would require to follow infection for longer time periods. But as I explained you before, this accumulation of debris inside the cells make this difficult to keep the cells viable for long time periods. So we also then infected undifferentiated enteroids. Those we can keep for longer periods in culture. And those enteroids, they are kept always in growth media. So we don't push them to differentiate and mature cell types. And here we saw that we have a higher viral titer that is achieved, hinting that the virus may prefer more immature cells than mature cells. Also, in this experiment, we tried to keep them for longer time periods in culture, and then we saw that the viral load remains stable up to 10 days post-infection, despite regular medium changes that are necessary to keep the cells alive, confirming that we have ongoing viral replication. We then attempted also to establish a transwell infection model. So in this system, in the transwell, in this setup, we have a control access to the apical and the basolateral side of the culture. So this is important when we want to follow, for example, the dynamics of HEV excretion through these polarized cells. And for this, we break the enteroids, 3D enteroids, into a single cell suspension that we seed in pre-coated transwells. We let them reach confluency, differentiate, and have a polarized epithelium. And then we infect it with the virus. We infect it apically to mimic the fecal or oral route that happens when a person ingests contaminated food. And what we saw was that the virus was mainly excreted apically. So these hinted that the gut could be actively secreting HEV particles into the lumen that are then excreted in the feces. Then our third method that we developed was the electroporation of single-cell enteroids with HEV capped RNA. So this is a methodology used very frequently in the field with immortalized cells. This is how we create HEV viral stocks. And after we electroporate these single cells, we plate them again in matrigel. Here you can see they won post-infection. We have our single cells. But over time, these cells, they keep proliferating, and we see that they regain their 3D form, that we structure as intestinal organoids. So these electroporated enteroids, they sustained an HEV replication over time and proved also to be the most the most effective method to reach higher titers of the virus. And here we are looking at the quantity of virus that was excreted in the supernatant of the cultures. But we also observed with immunofluorescence that we have an abundant presence of HEV capsid protein in these enteroids. And when we quantified this infection with high content imaging, we also see that we have an increase in the percentage of infected cells over time that corroborates also what we see in the supernatant. We, using this model, also further validated the activity of ribavirin. So this is an off-label antiviral frequently given to treat chronically infected patients. And because if the gut could serve as a reservoir for HEV, it's also important to understand if the antiviral drugs that are administered to patients, if they are also effective in the gut or only in the liver. Then, taking advantage that in the intestinal organoids we have multiple cell types, we wanted to try to understand, okay, which cell type is infected by the virus. For that, we did stainings with several markers for the different cell types, such as Ki67 for proliferative cells, sucrose isomaltase for enterocytes, chromogranin A for enteroendocrine cells, and the VGA that stains mucins of the goblet cells. And we saw that HEV infects both proliferative and mature intestinal epithelial cells. But we still wanted to see, does the virus has a preference for a certain cell type? So we quantified by high content imaging which cells were infected. And we saw that from all the cells infected, almost 2/3, almost 70% of the culture, they were proliferative cells, cells positive for the Ki67, showing a strong preference of the virus for this cell type. And as we saw also in the infection of 3D organoids, we got higher viral titers when we infected undifferentiated cultures. So these results were aligning. So we saw that with this increase in viral load over time, parallel also with an increase in the amount of proliferative cells during the infection, this suggests that infection of the proliferative cells is a very important and detrimental factor to have a successful and efficient replication of the virus in gut cells. But what we still needed to confirm was, is the virus that is being produced infectious? And also which type of virion is it, naked or quasi-enveloped? So we confirmed the infectivity of this progeny virus and characterized the type of virion using a density gradient centrifugation. That showed us that the particles that are excreted by the organoids are mainly naked. And these also show us that the secretion of naked virions that we find in the stool would come also apart from the gut, and not just the particles that came from the liver and mixed with the bile lost the lipidic layer. We also were able to see that we are producing a stock that is more infectious. We have here more infectious capsid protein than in any other system that is available. And when we infected naive 3D organoids with this viral stock, we reach a much higher replication when we compare with conventional viral stocks produced in immortalized cells. So these are all new features of the virus that we were able to discover using these different models. So each model gave us different aspects of HEV replication. They complement each other, but they also have their strengths and their limitations. So then we thought, how can we improve the physiological relevance of the model while also tailoring it to our objectives? That is, for example, we want to follow the culture for longer periods to understand how the virus is behaving. Can we mimic a chronic infection in vitro or not? So this led us to the next step of the model, that was the development of an organ-on-chip. So organ-on-chip systems, they are micro-engineered devices with microfluidic channels that replicate the tissue-tissue interface and also the mechanical forces that we find in vivo in the organism. And this tissue-tissue interface, they improve cellular communication and the functioning of the cells when we co-culture them, recreating what happens in vivo, because we have our intestinal epithelial cells, they are attached to an endothelial layer, laminar layer. So it's very difficult to mimic that in vitro, but with this technology and these chips, it's possible. Also, the mechanical features, such as the molecular gradients that are created, also the biochemical stimulation of the media perfusing, this really resembles the in vivo physiological properties of an organ. So in our case, we are using chips from Dynamic 42. These are commercial chips. And they have this microscopic slide format with two cavities, a left and a right cavity. Each cavity has an upper and a lower cultivation chamber that is separated by a semi-permeable membrane with a customizable pore size. In our case, we have 8 micrometer pore diameter to allow cell migration between the two chambers. And these chips, they are connected via standard Leuven tubings that can be then accopulated with a peristaltic pump. And the perfusion of the tissue with culture media, and it's independent for each chamber. So we can adapt the media and also the perfusion settings to each tissue to better resemble the in vivo properties that we see in the human body. So we are developing a gut on chip because this is we are studying the gut, and for that we are gonna put in one chamber human intestinal enteroids, our epithelial layer, and on the other chamber we want to create a vascular cell layer that will be composed of endothelial cells and macrophages. And with this gut on chip, we are combining the benefit of having this dynamic culture with the complexity of the organoids, and this will allow us to study, for example, dissemination of HEB beyond the gut. Does it infect the endothelial cell layer or not? These are all facts that we don't know yet. So as you can see here, we have like the timing and also the layers that are in each chamber. And for example, the cells, they need to be sequentially layered in the chip. But because each one of these cell layers, they require optimization before another layer is introduced, we are starting with a simpler gut on chip where we just have our intestinal epithelial layer with our intestinal organoids. And the first thing we wanted to see was what is the effect of this perfusion on the organoids, because we are used to do the transwells, we know how they look when they are polarized, but what is the effect of this media perfusion? So in one chamber, we kept it in static conditions, and the other chamber, we put the media in a circular perfusion. And when we look at the chamber that was in static conditions, we see a very typical polarized monolayer of cells, as we see in the transwell system. But when we look at the layer that was under perfusion, we see this spontaneous formation of the 3D structures, where the cells are kind of protruding out of the epithelial cell layer. So we are regaining these 3D interactions, cell-to-cell interactions that we lost when we break into a single cell and see the monolayer. And this was the only difference between these two chambers, was just the perfusion, just the shear stress that caused this spontaneous restructuration of our cell layer. So now we are also looking into the barrier function of the intestinal layer, also to which extent the cells are differentiated and which cell types are present. But we already started by doing some preliminary experiments infecting with the virus. And here you can see the experimental procedure that typically we need to do it for every experiment, where we start by sterilizing the chip, coating and then seeding the single cell suspension of intestinal enteroids. The media is changed several times, and when we reach a confluent cell monolayer, we connect the chip to the peristaltic pump and we put the media in circular perfusion. So after a few days, when we see this spontaneous formation of the 3D structures, we then perform the infection. And the infection is performed by putting the virus in perfusion together with the media for 6 hours. Then we wash the cell layer and we follow every day to measure how much virus is being excreted in the supernatant. And here you can see that, again, we see more excretion into the apical side than the basolateral side. So we are reproducing what we saw in the transwells, what also other groups described in literature using primary models. So we see that the model is working as we expected. And we see also a decrease in the viral load over time, as we also expected, because we are daily changing the media of the chip. But after 1 week, we see that the viral load is starting to rise again, indicating that the viral replication surpassed the loss that we caused by removing all the media every day, showing that the virus is continuously, slowly but continuously replicating. So now we want to extend these infections for longer time periods to try to fully characterize HEV kinetics in the tissue. So while we are working on this gut-on-chip and multi-layer gut-on-chip, we are also working in developing a liver-on-chip, because our final goal is to connect the gut-on-chip with the liver-on-chip to be able to study HEV infection dynamics between these gut-liver axes, and to try to understand and pinpoint, okay, in which step does the virus enters, replicates, or just crosses the layer, to try to get more information about this gut-liver axis, because these will have important impact in the treatment and the management of infected patients, especially the ones with chronic infection. I just want to finish by thanking the people involved in this project, especially Dr. Zhang. And thank you all for your attention. I'm happy to answer your questions. If later you want to discuss more about it, you can also find here the information. Thank you.
Thank you, Nanci, for your presentation. I don't see any questions in the chat for now, but I was just wondering, so you are working with enteroids, so I was wondering if you can also do it, why did you, if you work with a particular part of the intestines, because you have different, yeah, it differs. And so can you also make enteroids for other parts of the intestines? Would it have an interest for this particular virus?
Yes, for this particular virus, we don't know yet, because as I said, no one has studied the virus, this virus specifically in the gut. But for other viruses, such as norovirus, is also a diarrhea causing virus, we see that the virus is able to replicate in the different segments of the small intestine. And in our case, we have in-house lines from jejunal and from ileum. And we also have one that it was made out of different parts of the small intestine, so it's more a pool of different segments. But it's possible. It all depends on which part of the segment you take your sample, your crypt with the stem cells. Then you can generate organoids that will have special properties that we find on that specific segment of the small intestine. For example, more expression of receptors for a certain function, or more endothelial goblet cells. It depends. There's differences. So it's also important to study the virus in the different type of organoids to understand if there's a specific segment of the small intestine. Colon organoids are also possible. That we have never tried for hepatitis E virus, but as I said, a lot of things need to be studied. It's not known, but it could be that this virus also infects that segment.
Okay.
I also had a question because I was wondering, you mentioned that you take material from surgeries. So do you see this gut-on-chip model and maybe the link with the liver-on-chip as a tool for personalized medicine?
Yes, that is, I think I find it one of the best parts of being able to do this in vitro, because we can really replicate the patient in the lab. And this would be very important, for example, for chronically infected patients where the treatment is not efficient. We can take cells from that patient, take the virus from the patient, and then in vitro test new treatments or try to understand why the virus keeps popping up. So this, I think it's the beauty of these organoids, the tissue-derived organoids, is a personalized medicine.
Yeah, it's very nice prospect. Actually, I had one more small question, just out of curiosity, because you mentioned in your organoid model, you also incorporate the macrophages as part of an immune system. So I was wondering if there is a reason you chose specifically for the macrophages, or do you plan to also integrate other aspects of the immune system or not, and why not?
So, of course, just putting macrophages is a very big simplification of the immune system. But yeah, we needed to start with something. So we went for macrophages because they are also one of the most involved in the immune response to these infections. And also we have practice with them in the transwell system that we have also in the group, a colleague that co-cultures them with intestinal organoids. So they are more easy to also to acquire and then test. But ideally, we want to then incorporate different cells, and dendritic cells are also important. B and T cells that will require very high level gut on chip, but to start, yeah, we should always start with the more basic blocks, like we are doing now, just the simple gut on chip. Then we are gonna add the endothelial cell layer and then the macrophages. And then of course, from that point onwards, it's important to study the different immune cells to see if they will have a different answer to the infection. Yeah. But that needs to be done stage by stage.
Yeah. I see we have one question also in the chat from Julia. Would this system be suitable for cancer models and their treatment?
For sure. That's I think that's one of the main applications of the gut organoids, to be honest. We are in a very niche that is use them to mimic infectious disease, but the cancer organoids are, I think, one of the main applications, and for sure this is possible, and it's already done by several groups, I think.
Okay, so thank you very much for your presentation. If you still have questions for Nanci, you can still put them in the chat or send a mail afterwards. I already see a question from Paloma. How could you ensure that all cells are growing in optimal conditions as they are different types of cells? For example, in the last model, gut liver.
So also the advantage of this chip is that we will have the gut and the liver separated. They are not being co-cultured in the same chamber. So they will be, the tubings will be connecting one model to the other. And at the same time, we will have specific media for every cell type circulating in the chamber. Of course, when we, for example, we aim to connect the gut layer to the liver layer, we have media that will perfuse from the gut to the liver. That probably will require a lot of optimization to guarantee that we have a media that supports both cultures. But that's why I'm saying every tiny layer that you want to add requires optimization. But it's to maintain them happy.
Yeah, thank you very much for one final question from Elena. Have organs already been connected through organs on chip models?
Good question. That I know of, I have seen that for, not on organ on chip, I think, but for animals, I know these microfluidic systems have been used, for example, for liver, not liver, lung from pigs, but this is more in a transplantation setting. Yeah. Connection of real organs with the gut on chip, I'm not sure, or other organ on chip, but that also, in my personal opinion, we go back to an animal with an organ to connect with the in vitro chip that we are trying to make to not use the animals. That would have been a very good justification to use that, but that I know of, that's not been done yet.
Yeah, thank you for the insights on these questions. So if there are no other questions, you can still contact Nanci via mail or LinkedIn, as I mentioned, or you can send them afterwards as well in the chat. So, but now it's time for the second presentation of this webinar. So it's Eloïse Bouges, and Eloïse will give a presentation on patient-derived tumoroids to model rectal cancer under radiotherapy in an MPS system. Eloïse holds an engineering degree from the École de Biologie Industrielle de France, and her research focuses on developing and refining an in vitro MPS model of colorectal cancer using patient-derived tumor organoids. So this project aims to investigate the radioprotective mechanisms of biotic food supplements in the context of radiotherapy, with the ultimate goal to improving the life quality for cancer patients. So Eloïse, the floor is yours.
All right, I will share. Hopefully everything works out fine. Let me know if you can see my slides.
Yes, we can see them.
Perfect. All right. So thanks first for the invitation and the nice introduction. So today I will present you a piece of my PhD project that is going to be focused on patient-derived tumoroids that I'm using to model rectal cancer under radiotherapy and within a microphysiological system. So I will do this presentation in different parts. So I will first give some context before moving on to some methodology. We will then see some results, and I will conclude and give some perspectives. So first, for some context, Nanci gave already quite some nice context. So I will give context that is more related to my topic. So I'm working on colorectal cancer. That is the third most prevalent cancer worldwide. Things that are important to know in this context is that radiotherapy is one of the most common treatments that is used for this specific type, and more specifically for rectal cancer. The problem of such treatment is that it can induce resistance mechanisms that can have an impact on the treatment efficacy. Some other things that are important to have in mind when you want to work and model this type of cancer is the very specific physiology that it has, with notably a renewal of the epithelium that occurs around every 5 days, the presence of an oxygen gradient, and also an epithelium that plays a barrier function. There's already a lot of models that exist. However, most of those models are still using immortalized 2D in vitro and mouse models. And the limits of those models is that they don't give enough human relevance. So to bridge this gap, in this project, we're working on getting closer to a more human relevant model by using a microphysiological system that can also be called an organ on a chip. And in my case, I'm working with the CNBio organ on a chip model. So here on this slide, you can see a table that gives an idea of what it brings to the table using such a type of system. So it allows to get closer to human relevance, notably by the possibility of using 3D organs and also tissues that can be derived from patients. It allows to have a flow perfusion. You can also represent immune system in general and the presence of acute or chronic dosing. It gives high throughput, but also fast time to result. So for more methodology now and some idea of the workflow of my project. So we're working with what's called patient-derived tumor organoids that we keep in culture, and we then use them to seed monolayers to create a monolayer model. When we have this monolayer model, we're then able to put it under radiotherapy setup, so to represent the treatment that a patient would go through. And we can also transfer those monolayers in a microphysiological system that I will mention as MPS for the rest of this presentation. So the interest of using a monolayer model is to get as close as possible to the large intestine physiology, because here I'm working with rectal tumor organoids. And the first big goal of the project is to set up and characterize our monolayer model. So you can see here on the left the general representation of the large intestine physiology. So as I mentioned before, there is 3 main components. So you have the epithelium that is here, separating the lumen, where you have the presence of your gut microbiota, and the lamina propria. There is also presence of an oxygen gradient that is here. And in the case of our monolayer model, the idea is to represent the separation between the lumen and the lamina propria with our monolayer that plays a role of a barrier, so the role of an epithelium. We are then moving on to the next step of the project, that is working with irradiations. So the goal is to represent the treatment that a patient would go through. And to do that, we are working with fragmented irradiations that allow to have an idea also of a dose response so that we can target an optimal therapeutic window. An optimal therapeutic window would be described as the dose that would create enough cell death for the cancerous cells, but that would not impact the healthy cells on a too high level. And then for the last part of the project, as I mentioned before, I'm working with organ on a chip barrier plate from CNBio with the idea of implementing our monolayer model within such a system. So it allows to have an aerobic basolateral flow that you can see here on the little video on the left. And now we can move on to some results. So first, I will talk about the monolayer characterization and some images to have an idea of what it looks like. So Nanci already gave quite a good overall idea of what's an organoid, but I will go back on it very fast. So what's an organoid? It's a multicellular 3D structure that allows to maintain tissue morphological characteristics, and it can be patient-derived, and also it can be generated from tumor tissue, which is the case in my project. So the first step is working with organoid subculture. so that you can then use them, fragment them, and seed monolayers. To have a little idea of what the monolayer looks like and how it's forming. So you can see here on this slide what it looks like from day 0. So basically after the fragments are seeded on the transwall, and how they are forming, slowly reaching confluence before we can use them. To have a bit of a closer look on what they look like, you can see here first on the left a bright field image that gives an idea of the general structure of the monolayer. So you can see that it's a very tight monolayer, and it only gives a very general idea. But if we want to have a bit more information on how the cells are linked together and also of the morphology of our monolayer, we also used SEM images. And we can already see that our monolayer is not completely flat. You can see that there is some kind of topography that is present. But this is also visible on images that I will show a bit later. To also have a bit more information of the characteristics and the physiology of our monolayer, if we go a bit closer with our SEM images, we can see that on the surface of our monolayer, there is the presence of microvilli. So basically, each pinhead that you see here on the right is one microvilli. To have a bit more information on the structure of the monolayer and how the cells are organized together, we also did some immunofluorescence imaging to have an idea of the cytoskeletal integrity and also the organization of the cells together. So here on the left, you have a cryosection. So you can have a look at the monolayer from the side to have an idea of how the cells are organized. And you can clearly see here that there is a polarization of our monolayer with the nuclei being located on this side where it's the bottom of the transwall. In the right here, you can also see an apical view of this monolayer, just to have also an idea of how it looks from that point. One tool that we use to assess how the monolayer is forming and also more specifically to assess the barrier integrity is the tear measurements. So we are using this type of device where basically the goal is to measure the electrical resistance that there is between the two sides of the monolayer. So it allows us to know when the confluence is reached and to have also a validation of what we are seeing in the bright field images. So here you have 3 different experiments that are shown. I put them here because I wanted to also point out that the seeding density is very important and is going to have a huge impact on how your monolayer is going to form and when the confluence is going to be reached. So you can see that here in this first concentration, there is a very slow progress from the start, and that then after some days, there is a peak of the values that then reach a plateau that indicates that the confluence is reached and that it's not evolving anymore. While when you use a lower density, you see that there is a very, very slow progress that can also have an impact on your experiments later if you cannot reach a plateau. We can see here also with the higher density that you have a peak that comes way quicker, but that then there is some kind of drop in the values. This could also be explained by a possible maturation of the monolayer itself that might differentiate and have some changes in the tear measurements. I will now move on to the radiotherapy setup part with first an idea of what the setup looks like. So as I mentioned before, we are working with the chronic treatment. So we're doing 5 different irradiations on 5 consecutive days. So here are the doses that we've tried so far. And for a bit more of a visual idea of what the experimental setup looks like, here are some images of the beam that we're using. So basically you can see here on the left that we put a hit plate here on which we put a kind of target with the plate on top of it. The beam that is irradiating is this part. If we look a bit closer, this is what it looks like. And the white part that you're seeing here is actually called an alpha film. So this is the image that you see here on the right. Don't take into account this white strip that is an artifact, but basically the idea of using an alpha film is to have an idea of what is receiving the dose. So it's a nice tool to assess where the dose is spread around, is it even? And also it gives an idea of what really received the dose. So here you can see the outline of the plate that received, and also you can see the transwalls, and the medium that received the irradiation. So to have an idea of the impact of the irradiation, we looked at permeability. So again, we used the tear measurements that I mentioned before. And in the irradiations that we did, we noticed two different significance of an impact and a decrease in this barrier integrity after 8 and 10 grays. And another thing that we looked at to assess the impact of the irradiation is the impact on the cell viability. To do that, we used LDH assays to have an idea of the cytotoxicity and the level of it. So far, the only dose that created a significant response was 6 gray. So it's also kind of going with what we see with the tear measurements, that there seems to be a resistance happening of this specific cell line that we're using. Another thing that we looked at is through proteomics. So we looked at the impact of the irradiation on the proteome in general. So here are some volcano plots that give a general idea of how the proteins that were detected are spreaded. So you can see that there is a lot of proteins that were detected but not differentially expressed. And you also have some up and down regulated proteins. Here are some Venn diagrams that give maybe a bit more of an idea of the number of proteins that were detected. And also we can see that there is a bit more of protein that were detected and significantly differentially expressed when the dose is a bit higher. To go and look a bit more at the pathway level, here are some heat maps that give an idea of the proteins that were up and down regulated in specific interest pathways. So on the left, you can see programmed cell death. And the first conclusions that we draw from those results are that it seems like the cells are creating a regulation of the apoptosis, and it's highlighting an early and also late apoptotic signaling that is as a response after the irradiation. At the level of the cellular response to stress, we can see that it seems that there is some mitochondrial and autophagic roles that are being up and down regulated that might be a response as a mitigation of the irradiation. And on the right, you can see the pathway cell cycle mitotic with what seems to be an emphasis of the disruption and the regulation of cell division. This is a very important thing to take into account, as the cell cycle are very crucial for the amount of DNA damage that is going to be present and visible. And now I will move to the implementation of our model in the microphysiological system. So first, for some idea of the experimental flow. So basically, this is the same process as the monolayer model that I mentioned before. The main difference is that we seeded our monolayers on the other side of the transwall. So on this part of the transwall, we just seed it there and then switch it back into our well. We grow the monolayers for 2 weeks and in a static plate, so without any difference. And then when they reach the confluence after 2 weeks, we transferred the transwalls into our plate that goes into the MPS with a flow rate of 0.5 microliters per second. Again, we checked the tear measurements to have an idea of whether or not adding a flow perfusion would create a stress or if it would have an impact on the integrity of our monolayers. We noticed that after 4 days of the flow, there was a significant decrease of our integrity. This still needs to be repeated to be able to conclude on it, but it gives already a nice idea of what is happening and also what should be taken into account for later tests. Another thing that we looked at to characterize what the difference of adding a flow creates, we checked at the morphology of the monolayer. So with those cryosections that you can see here, we can already see that there is a difference of thickness of our monolayer. We calculated the average of those thicknesses, and we noticed that there was indeed an increase of a significant increase of the average thickness of our monolayers. There is also the presence of such types of surfaces around the monolayers. As I mentioned before, when we looked at the SEM images, where I was saying that for sure the monolayer were not completely flat and that we were seeing some topography. This is also another way of looking at it. We see it both in MPS and static, but it's thicker and more present in the MPS system. Another way to look at the difference that using MPS can make is the mucus. So as we're working with colorectal tissues, we know that it's the different types of cells include goblet cells, and goblet cells produce mucus. So one way to check it is by staining it with immunofluorescence, as we did here. So on the left, you have the MPS square section, and on the right, the static one. And we used a stain to look at first the cytoskeleton, the nuclei, but also the presence of mucus with Mus2 stain. So we can see that it seems to be present in both of the models. However, it seems to be more present in the MPS. I will now move to the conclusion. So as we mentioned, we in this project are working on setting up and characterizing our monolayer model. We want to assess the response to irradiation and also implement our monolayer model in an MPS. The goal of this project is to go towards personalized medicine and with the idea of improving patients' quality of life that go through radiotherapy treatments that are very harsh treatments. The perspectives of this project is that it allows to model and to represent the rectal cancer with as much human relevance as possible under radiotherapy. This gives also a platform that allows to assess radiosensitivity, and that can also allow to test radioprotective agents, for example, and radioligand therapy. So now I will conclude on this. Thank you for your attention. I wanted to acknowledge all the people that are supporting me in this project and participative in this project. And I will now leave the floor for some questions.
Thank you very much, Eloïse, for a nice and interesting presentation. I think everything was very clear. It's also fascinating that you work with radiation. I actually have already a question because I was wondering, because you nicely implemented with the personalized medicine and the improved quality of life. Like, do you know like a timeline which it could be implemented, the technology that you're using or the setup?
You mean working directly with patient samples?
Yeah.
For a timeline, I wouldn't really be able to tell you. I'm already working with samples that are from patients. In my project, I'm not working with a hospital that would provide samples, but I know that some groups are doing that. So I know that it's already a possibility, but just at the level of what I'm doing, I could already see how interesting it would be, notably because with the radiations that I'm doing, I'm noticing that the cells are not responding by just dying after being irradiated. So it seems that there is some kind of resistance, and this can be very nice to know ahead for a patient, so that, as Nanci was saying, so that you can like take samples from the patient, because normally when a patient is diagnosed with rectal cancer, they don't go straight to radiotherapy. There is first surgery, so you have samples from the patient. There's also chemotherapy that can come before radiotherapy. So there is time also to test the samples and check how they would react to the irradiation, so that we can know, okay, what's the best strategy for the patient? And are we going to create so much secondary effects to the patient while we already know that they're super resistant? So I think it gives very nice information to also the doctors that would be able to choose the right strategy.
Yeah, indeed, I think it would be very useful for the doctors and the patients as well, that they could be protected a bit for the side effects of maybe unnecessary radiotherapy. So I also
have a question. I was just wondering, because you talked about the optimal therapeutic window, and you said that the ideal is that it's killing cancer cells, but not hurting too much the healthy cells. So can you test that also in vitro? Like, if you then would you need to have healthy cells incorporated?
Actually, this is like, for me, of course, models could always be more perfect and more representative. If I could have access to both healthy and cancerous tissues from the same patient, it would be awesome, because I would be able to treat them the same way. I would, of course, not put them together in the same transwells. I would do them separately. But it would be really great to have this response of both, so that you can actually assess the response of both. In my case, I only have access to tumor organoids, but then also you have to take into account a lot of things for radiation treatments. You have to take into account the dose rate that you're using, so how fast the irradiation is being received. This is also a possible perspective for my project, of testing another dose rate to see if it would create a stronger reaction or if it would be the same. But yeah, it would be like, I think it's also a part of the project is taking into account the impact that it has on healthy cells, because of course, when you're irradiating the pelvic area, you're not just targeting the tumor.
Yeah. Okay.
Good point.
Thank you.
So I see no more questions in the chat. So that leaves me to thank you, both the speakers, for their interesting presentations, and also to you, the participants of this webinar. After this webinar, you will get a survey in which you can score the content, and also where you can put suggestions for other topics or speakers. The people who indicated in the chat that they want a certificate of attendance, they will receive it within 1 or 2 weeks, a bit depending on the time schedule. Also, just so you know, the webinars are recorded, and they're also placed on our YouTube channel, so you can rewatch them later if you are interested. And then finally, I also wanted to invite you to our next educational webinar, which will be on the 18th of December, where we will focus on pain on chip by a speaker of EMAC. And also 2 experts from Cienzano will give more information on applications for the monocyte activation test, which is an alternative method that was recently also included in the pharmacopée. So obviously, we hope to see you there. So thank you very much again for being here, for joining this webinar, for presenting, and also for posing questions. And we hope to see you within a month. Thank you.